Next up is Neil McGregor, who's joining us from the University of Melbourne.
Well, it's a delight to have Neil here as well. I met him in Florida at one of the meetings, but I didn't realize he had worked on ME CFS for a long time. And it's great to have people that have been in this field for a long time. But I wasn't sure he would come from all the way from Australia, but he graciously said, yes, absolutely. So it's great to have him here, a wealth of knowledge, and thank you very much.
Good morning. Good afternoon in Australia, or good evening maybe. All right, so what I'm going to talk about is, after my 25 years of looking at this sort of data, I mean, I've got to learn that CFS people don't have all the same symptom outcome. They might have similar onsets, and when you look at them, and you do factor analyses, you find out there's a depressive set, and you find there's a pain set, and you find there's a set with lots of sort of taste and smell and symptom exaggeration such as that. So there must be some explanation for that. And I'll get the right button in a minute. Sorry, first of all, I better give a list of all my collaborators, and they're all listed up there. We have a set of scientists in the top two rows. Most of us are involved in the Bio21 Institute, and then we've got two clinician groups that provide us with patients. And they're probably the most valuable of all, because if we can't collect patients that are appropriately qualified, classified and all that, well, we don't have anybody to study, which is a bit of a problem I've noticed around here, isn't it? Yeah. So after all, analysing all of that data, what I realised is that, and there are studies out there now that show that we are actually dealing with what appears to be a heterogeneous event, not a single thing. We define them according to a symptom set, and that basically is, we define them predominantly on fatigue. So if we analyse fatigue as a thing, what we find is that there's clusters of things that go with that. We find that something like 50% of the patients may in fact have pain syndromes or fibromyalgics, but the others don't. There's a whole cluster of them that are cognitively impaired, but the others don't seem to have that. And it's a bit hard to see this writing down here, so I apologise. So what we really want to know is, why are these different? Is there if they have a similar onset and a different outcome, that can only be explained by a difference in their genetic response, or some change to pathogen or other epigenetic types of issues that may actually drive the event. So if we do some simple regression analyses, like in other words, we use a sophisticated statistical technique to look at how the patients score their fatigue scores, and then we look at the metabolome, what we find is the predominant thing that we see in our data set, or the first of morning samples, where we actually find the glucose are elevated. Probably we did them postprandial or after meals, we will get some variation in that. And that seems to be the metabolome ones from over here seem to have, because they're not restricted in the same manner as we do. If we look at fatigue, what we find it's related to a glycolytic change, and it's related predominantly to the glucose level in the serum. But if we turn around and then we look at the ones with fibromyalgia type pain syndromes, and we look at the amount of widespread pain they have, we find that it's quite a different event metabolically. We see there's a change in kidney function. We see that they're, during these, when the pain is worse, they've urinated out more amino acids, they've lost more electrolyte, and we can actually show that. The problem with seeing that in chronic fatigue syndrome people is we only see them after 6 months, because that's what the definition tells us. But because we've actually seen other pain syndrome clusters at their time of onset, we actually see these quite a different set of changes, and when you put them together you can actually understand what the relationship is. So basically what I'm going to try and do now is look at the genetics of it, and to see whether we can tease out sort of things that may indicate some of these sorts of things. So we're currently doing a pilot study, so it's unpublished data, so nobody's going to tell anybody, is that right? Good. So what came out of that is that we took 56 patients and 32 controls. We used a data set. I think the Kenny DeMerlio's study used a DNA chip chasing immune changes. What we've got here is one that just changes general chemistry. So they're quite different sets, but what we ended up with was 316 SNPs that were, or mutation points that were different. Of those, 162 were in known genes or protein production. 16% of them were in what we call RNA genes. These genes actually regulate, or are known to regulate, how the DNA is transcribed down to a protein, usually in the cell down to the messenger RNA, which exits from the nucleus. So if there's anomalies in those, or there's viral interference with, where they make a small protein, it may interfere with that process, and that may alter the transcription of the final proteins. We also found that 33% of them were potentially junk, according to the old definitions. But what we also now know is that some of those potential junk DNA actually do things. And we can see that they act as promoters for genes. We can see that some of them may actually have enhancing functions. So what they do is when they get activated, they will enhance the production of a particular gene. Now, one of the problems we noticed with a couple of studies is that because the control groups are so small, the important thing was for us to make sure that our control group wasn't biased. So, in other words, we went to the 1,000 genomes data set and we compared every SNP of everything that we had and looked for any of those that where our control group may be aberrant, or if our CFS group matched the normal distribution, all of those got eliminated. So we basically ended up with... So the other thing we did was we removed any of them that didn't have what we call an odds ratio of 2. So in other words, they were more than twice as likely to be present in the CFS group compared with the control. So if they were less than twice as likely, they also got eliminated, because that means they're probably not as important. We can go back and look at those later, but the important thing is that we don't get biased in the beginning. So we ended up with a data set of 38 genes, which is not a lot when you actually think about it, out of the huge lot that were potentially there. So the first thing we did was we wanted to see, is there relationships between the genes? So what we did was we did factor analysis, and I did what I call layered factor analysis, where you divide the group into 2, 3, 4, 5, and you look at what products and combinations come out of that. And what we found was there was at least 7 major clusters. The first one was an anomaly in G protein couplet receptor proteins. The second and the third ones were the same combination, but in combination with a couple of other or additional genes. So whether these other genes were probably amplifiers, modifiers, or things that may relate to symptom expression, they were suspicious that's the case. Then the fourth, fifth, sixth, and seventh all involved RNA helicases. Now what these do is they remove viral and bacterial and our host RNA out of the cell. And you'll see that some of those had some additional genes that they were present with. So when we just looked at a couple of the individual SNPs that were there, and what we found is some of these had around 5-fold more likely to be present in the chronic fatiguers. And down the bottom, some of them were present in heterozygotes. In other words, they may be autosomal dominant, and others were present in the homozygote state where they're more likely to be autosomal recessive. And we can see the G proteins, and the last lot is a thing called Langerin, and I'll talk about that in a minute. So what we just simply did was we took all the mutations within the genes that had low prevalence within the community. We just did a simple addition. We then did a simple addition to find out how frequent they were, and what we found is that the Langerin was 3-fold more likely, the G proteins were 21.5-fold more likely, and the RNA heliaxes were 12.6-fold more likely. Anybody with an odds ratio of 2 as a scientist is usually pretty happy, but when you get bigger ones like this, you're quite ecstatic sometimes. A G protein basically is constructed out of 3 components: alpha, beta, and gamma. And there's something like there's 20 alphas, 5 betas, and something like 12 or 13 gammas. And different tissue distribution of those determines how they respond in various tissues. What basically happens is when the receptor gets activated, the beta and gamma separate. The alpha and the beta and gamma separate, and they go and perform separate functions. And we look at what G proteins do. What we find is that they affect taste, smell, mood, inflammatory issues, blood pressure, heart rates, water balance, electrolytes, and visual. Now, if we start thinking about our ME CFS patients, we can see lots of people with lots of those sorts of things. So I think we're beginning to think, or at least I think, that some of these may be critically important in symptom expression of the type we see within the symptom cluster, within the cluster. Very quick one about RNA helicases. What they do, in essence, is they remove double-stranded DNA, viral RNA, host RNA out of the cytoplasm. They are inhibited by viruses joining to the epidermal growth factor, and what they basically, these potentially could be the major reason for the viral triggers, where they interfere with if we're not able to remove the viral RNA. The Langerin or CD207 gene, what it is expressed on dendritic cells in the gut, on the skin, in the throat, and basically it is a receptor for pathogens. So if we have problems in this, we're going to have particular problems in detecting certain viruses, etc. What we know with HIV studies that those where they've actually inhibited Langerin, they've had greater viral loads, and they've had more infections within the body. If we look at the viruses that can be transported or transmitted or acquired by this receptor, we see HIV, HSV1, HSV2, EBV, in other words, a long list of ones that we know interfere with CFS patients. We also did an analysis on the mitochondrial DNA. We found nothing. We found one mutation that indicated haplogroup H5, and when we cross-checked that to see whether it was a mitochondrial anomaly, what we actually found was that all of those were carriers of the Langerhans mutation set. So we think they just happen to be, they may actual fact be an indicator of a line of transmission of that particular gene group. So in summary, what we've got is, by using a sort of a slightly different approach, we have been able to identify a number of potential genes. There's certainly a gene cluster associated with G-coupled proteins. There's a gene cluster related to RNA helicases, and there's a gene cluster related to Langerhans. Now, clearly, this data may be nothing. We have to make sure that this makes sense and it's not no sense. Thank you.
Thank you so much, Neil. Thank you, Neil. We'd now like to welcome up our next speaker, Alan Light from the University of Utah. Sorry, you'll go one soon.
I've known Alan for quite a long time. I have a lot of friends at the University of Utah, and it's a pretty underrated university because it has some really very elegant and great researchers there. And you're going to hear another one, Waldemar Oliveira, as well, from the University of Utah. I found that Alan was really working on other aspects of this disease. He's an expert in pain and those kinds of things, and brain interaction. And so I'd like to have him be involved, and that's why he's gotten involved in this as a collaborative effort. And he's not very far away. He doesn't have to come from Australia or Sweden. It's a short plane ride. So thank you, Alan.
Thanks, Ron. So what I do is quite a bit different than everything else you've been hearing so far. So the first thing I'll do, of course, is sort of introduce the group here. So Kathy Light, believe it or not, we're only related by marriage. So, but in fact, she does most of the work, and so I'll attribute almost everything to her. And she gets it right, I get it wrong almost every time. Dr. Cindy Bateman is our clinician extraordinaire, and that's true. And many of you know about her already, and she is just absolutely fantastic in terms of, we could not do any of this work if it hadn't been for her. Andrea White is our exercise scientist. She also helps us with a lot of other aspects. Marcus Aman, I'm going to introduce you to him for those who don't know him. So he is actually also an expert in fatigue, but he's actually been interested in fatigue from the point of view of true fatigue, not chronic fatigue syndrome. So what the heck is fatigue at all, period itself? And that one actually is very important to really understand what's going on in chronic fatigue as well. And then Ron Hugan, who is our lab organizer, and without him, again, none of this would have happened. In addition to things that none of it would happen, I have to thank our Department of Anesthesiology, who has been very generous in actually helping us with funding a lot of the studies we've done. The University of Utah School of Medicine also contributed early on to our work, which was really brave on their part. I don't think they knew, I didn't know for sure, when we first started working on chronic fatigue syndrome, how controversial it was, but they gave us a good pot of money to get this started, and Cindy was one of the people that benefited from this early on as well. And then, of course, Solve CFS, the American Fibromyalgia Society, and then not a whole series, but a few very, very helpful patient donors who made much of the research that we're doing possible. Okay, pain and fatigue. So first of all, I'll let you know a little bit about, for those who don't know about me, is that I'm a neuroscientist. I'm not a metabolomic scientist. I've been doing research on the basic mechanisms of pain for over 40 years. Did most of this at the University of North Carolina, moved to Utah in 2013. And really, for 40 years, I've been wanting to actually work on fatigue. So 40 years ago, there was a debate about whether pain was actually a specific sensory system at all. And it had many of the same problems that fatigue has today. Essentially, our group, led by Ed Pearl, who died just a couple years ago, proved that it was both. It was a specific sensory system. There were specific neurons dedicated to evoking pain, and also that it had emotional aspects to it, and all the pathways have been solved since then. And like fatigue today, pain was at that time considered to have no objective features, and that we actually showed that showing that the fact that there were specific sensory pathways ended the arguments about whether was this something that we could actually really work on, could we do anything about it, or was it something we should just give to the psychologist, not even the psychiatrist, because we had no treatments that would actually be helpful in terms of long-term pain. So all of this eventually led to strong science that has finally, only in the last 4 years, believe it or not, now we're coming up with effective treatments for chronic pain. And it took that long. I'm really hoping it doesn't take that long for fatigue. So, okay. So 11 years ago, Dr. Edwin McCluskey actually determined the key that made it possible for me to actually understand what fatigue really was. What he found was that the receptors for fatigue don't respond to a single signal, which is what we had thought for many years. Instead, they respond to a combination of signals. And those signals are exactly the same substances you make when you exercise, as it turned out. The things like lactate, well, yeah, that's there. Protons, meaning it's acid, that's there. And also ATP of all things. ATP actually comes out of cells, as was shown previously. And when you actually exercise, it doesn't stay inside. It actually comes out when you exercise. But all three of those need to be present simultaneously for you to actually feel the sensation of fatigue. Okay, so that's the way it really works. So from that, and it's actually my wife again that decided we should do this. We were using mouse models at that time, and she convinced me that, wait a minute, mouse models aren't that great when you're talking about fatigue, because you can't ask the mouse, are you running because you're fatigued, or do you stop running because it's pain, right? So, and she's right, of course, but I said, we can't do this. And she said, yes, you can. And as I've said, she violated the IRB and said, well, I'll be the first person to try this out. So she was, and whoops, what did I do there? I shut the whole thing off? Oh no. So how do you turn that back on? What did I hit, the off button? Yeah, this is, yeah, sorry about that. Oh, it's on there still. I just turned I did turn the whole thing off. This is like Spaceballs. Well, you turned the whole thing off. Okay. Have to be very careful when I push the button here. So at any rate, let's see how far did I get through? Oh, yeah. Let's see. So it turns out that we eventually discovered that fatigue was, in fact, part of a complex system that protects people from using up their energy stores. And this system includes parts of the cardiovascular system, the autonomic nervous system, and the sensory receptors that influence nearly all parts of your body. So fatigue, that's fatigue. It's not chronic fatigue. That's normal fatigue. That's what it's supposed to do. I did it again. Why is that doing that? Is it, which button it is? It's the vision, this one right here, yeah. No, the thing is that I guess I have to really be careful. There. There we go. Okay. So, well, yeah, it's probably not a bad idea. Although, at this point, I think I got this down. So, if I didn't, I'll be sorry. So she convinced me that we could use gene expression to actually look at this in humans, and she was the first one that actually we showed that gene expression really did work in terms of looking at fatigue itself. Okay. So again, the fatigue we were looking at is not ME CFS. So, and the fact that our gene expression is from immune cells suggested that immune function might actually hold clues to the causes of ME CFS and fibromyalgia. So when we actually use the gene expression in our ME CFS patients, we found that in fact there was a very large difference in the gene expression in at least 60% of the patients who had chronic fatigue syndrome. We published two major papers on that. The last one in 2011, and in fact, the problem is, of course, there is another subgroup in that in fact, instead of having increases in gene expression, they had great decreases in gene expression, which is part of the reason other people haven't actually been able to see this very effectively in their studies. You have to actually have the right patient population to actually see that. Right, here we go. So the fact that the expression in the immune cells it gave us an idea that there might be a clue that fatigue there's something special about fatigue that in fact might be something a little different than we thought it was. And that clue was that fatigue is common in mitochondrial diseases. So now I'm going to talk about autoimmunity, mitochondria, and mutations in ME CFS. And the question was how to put this all together. How do those things combine? How to get autoimmunity and mitochondria mutations? How does that have, what's in common here? Well, there's been a couple of recent publications that indicated that autoimmune disease might cause chronic fatigue syndrome in at least a subgroup of the patients. Two of these papers suggested it might be treatable with drugs that alter autoimmune response. And so and one of them was Janusz. Janusz, okay, I get it wrong. So I knew I was going to get it wrong. Sorry about that. Correct me. No, he's not going to correct me. Okay. Yes, Dr. Berquist, there we go. That's the way to do it, okay. So at any rate, we collaborated with Dr. Madeline Cunningham at the University of Oklahoma. We gave her blindly samples, and she gave us some findings on what really happened in some patients. Okay. This pilot study, altogether we found that, and this is what she found, basically. She sent the data back to us, that 15 of 18 patients who had chronic fatigue syndrome had positive autoimmune findings to the beta-adrenergic receptors or muscarinic receptors. Now, that's exactly what Dr. Berquist also showed, and that which was fairly, when we saw it, I hadn't seen it till just this week, those results, but that was very comforting when we saw that. These receptors normally control the flow of blood in muscles and brain. So that's one thing to know, of course, that it would obviously cause many of the symptoms that the patients here have, if in fact you had those autoantibodies. Well, these autoantibodies could cause the symptoms seen in these patients. The question is, why do these patients make these autoantibodies at all, right? So why should they make these? Well, this is sort of our current hypothesis and a very popular hypothesis, and that's a thing called molecular mimicry. So the molecules are mimicking something. Normally, the immune system fights off diseases like the flu, mononucleosis, strep, and those sorts of things by making antibodies that specifically target certain proteins in these pathogens. However, these targets often, maybe always, are similar to proteins in patients that are found in the fatigue system. Remember, I told you it's a complex system with lots of vulnerabilities. So if you target any portion of that, you could get the phenomena that the patients experience. So this may be why these diseases also cause fatigue as one of their symptoms. So the fact that, in fact, they are attacking the fatigue system. So normally, after viruses or bacteria are eliminated by these antibodies, in this case not autoantibodies, once they are quit, we get rid of those, normally the immune cells quit making the antibodies and the fatigue should go away. That's the normal thing that happens. Whenever you get sick, everybody gets fatigue. But then when you get better, it goes away. I think that good explanations also came for that same phenomena from a talk earlier today. However, in some people, the immune cells don't quit. They keep making autoantibodies against the fatigue system. When these people then get re-exposed to what the antigen was in these pathogens, they get even more fatigue. So why don't immune cells quit in these people? That's the big question. So we hypothesized that this may be because they have a mutation in some other genes that are causing the immune system to misbehave. So we decided to use RNA sequencing of immune cells to find both genetic mutations, inherited mutations, and mutations acquired later in life that might cause immune cells to misbehave, causing the fatigue. So we focused on genes that could affect the immune cells that make autoantibodies. We also focused on mitochondrial mutations because there are a lot of them. And this is also because, as been explained, mitochondria are the energy source for all cells in the body, and they also influence autoimmune responses. There are actually several autoimmune diseases, and I've pointed them out there, that appear to have a mitochondrial contribution. So what's the connection of mitochondria to ME CFS? Well, I'm going to skip this part. This is a mitochondria. You already saw what it looked like earlier today, and there was a lot of discussion there already about what could happen with these. So we have minimal findings so far, and I'm only going to tell you just a little bit of the data from our data from 45 patients that had CSF or FM and 31 controls. So what we found is that nearly all ME patients had mutations in mitochondrial genes that weren't seen in healthy controls. So this is different than Dr. McGregor's study. This is RNA-seq, so we're only looking at immune cells here. And we're only looking at cells that are expressed, that actually are doing something. So some of the patients had many potential fatigue-causing mutations, but not all the patients shared the same mutations in the same mitochondrial genes, which suggested that there are different subgroups in the ME CFS patients. So nearly all parts of the mitochondria had mutations in at least some of the ME patients, indicating that the specific location of a mutation might not be important in causing ME. If we look at the chromosomal genes, we found that so far, no mutations were specific for ME in genes involved in autoimmunity. We didn't find any of those. Instead, we found one mutation in several autoimmune disorders. So there seemed to be general for autoimmune disorders, not for ME. We found that this in 28% of the patients. These gene mutations, we also found gene mutations involved in steroid receptors, including estrogen, which were found in as many as 25% of the patients. 12% of the patients shared a significant mutation also in the heat shock gene, which many people have said might be important in fatigue as well. This gene is required, as some of you know, as it's necessary for the proper production of a lot of different proteins. Okay, in summary, fatigue is a complex system involving cardiovascular, autonomic, and cognitive components. It's a complex disease that likely involves an autoimmune dysfunction in at least some of the patients, along with possible mitochondrial contribution to the dysfunction. At least some cases of chronic fatigue syndrome may be caused by a combination of multiple mutations in mitochondria and chromosomal genes. We now have samples from more than 280, and we'll soon have 340 patients with chronic fatigue syndrome, but that we're actually comparing with those who have ME and fibromyalgia, as well as depression, migraine headache, and also healthy controls. And we're doing RNA-seq on all these to look at the mutations and gene expression as thoroughly as we can. This includes a number, in this, we're including a number of families, because it turns out that there is a familial relationship in a lot of patients that we, much more than we actually originally anticipated, that may have other autoimmune diseases. We've seen that there's very often a patient who has chronic fatigue syndrome. There are many members of their family that also have different kinds of autoimmune disease.